Hello, everybody. Yeah, it's a really unbelievable pleasure to welcome Professor Dr. Andreas Horn today, yeah, for our lecture series. So Andy started actually medicine in Freiburg, and then he moved to Berlin. There you did already a doctor, yeah, Dr. Med Or in Freiburg.
Yes.
Yeah, and then he joined here like the medical, what was it, like a clinical
medical neurosciences.
Medical neuroscience. And then the great thing was that he actually worked together like in my group. Yeah, so like we did a wonderful work. Andreas was developing a full brain connectome, yeah, where he combined a structure or DTI imaging with default mode network. Really amazing work. I guess maybe he's also telling us a bit about that today. And from there, he then decided to go on with his clinical education and joined the neurology department at the Charité, where he worked together with Andrea Kühn. Did a lot of like deep brain stimulation, and there he combined both interests, like the clinical one and his research with us, with the connectome, and developed a really nice tool like the lead DBS, a tool where you can actually see exactly where the deep brain electrodes are and what kind of like structure they are affecting. And this toolbox has been used by hundreds or thousands of researchers worldwide. And then he got also like a pretty nice grant, like a Heisenberg professorship. But in addition to that, also like the offer to go to Harvard, the neurology department there, in order to become a professor. What he actually preferred in the end, unfortunately, he's now in the States, but hopefully coming back at some point to Berlin. And it's really nice having you, and I'm very much looking forward to your talk.
Thanks so much, Felix and Timo, for the nice introduction and for this invitation. It's really great to speak to you, and I try to make this as interesting as possible for a more general neuroimaging audience. But I will indeed also speak about neuromodulation, and this is just a slide about the different concepts of how we interact with the brain, not with tasks, but with actually stimulating the brain or modulating the brain. There are invasive ways and non-invasive ways, and specifically all of you will be familiar, of course, with most of these things, but non-invasive neuromodulation includes transcranial magnetic stimulation, or then also the more electrical transcranial concepts like TDCS and TACS. And there's also, I think, TIL does that quite a bit, or did that quite a bit with LIFU, low intensity focused ultrasound now. So I want to say that I think a lot of what I'm going to show could be applied to exactly these concepts, and I've worked on a few TMS papers as well in the same realm, same concept. And then I'm talking a bit more about invasive neuromodulation techniques, and mainly deep brain stimulation. So this is a surgical technique where electrodes are being implanted into the brain. And then other forms of invasive neuromodulation could be seen as actually lesioning the brain, burning small holes in specific sites, which I won't talk a lot about, but also work with in my routine here. And so since there is quite some focus on deep brain stimulation, I want to spend 2 slides on briefly mentioning what it is. I already mentioned it's a neurosurgical technique. So electrodes are being implanted into deep structures of the brain by a neurosurgeon. There are burr holes being drilled, and then the cables run to something that looks very much like a pacemaker for the heart, which stays there for a long term, often 18 years, 20 years, essentially until death, and will emit these weak electrical pulses to specific contacts in the deep structures of the brain. And this is a patient video of an optimal case, just showing the same patient without any medication, so any dopaminergic medication, Parkinsonian patient with The only difference really being the electricity off and on the left and on the right. This was one of the really best patients I saw and treated myself in my time at the Charité, and this was the same moment I took the video, so it's really the only difference there is really electricity on or off. And you can see a marked difference here with movement speed and alternating movements and a lot of bradykinesia, also a little bit of tremor. Also in walking in a second, you would see that the patient would have a very normal walking gait. And if the electricity is off, you see a classical Parkinsonian gait. So what is interesting from a neuroimaging perspective now is that the electrode placement matters a lot. So it's really about millimeters here. These are 3 different electrodes from 3 patients, just exemplarily shown. And this is the optimal hotspot that we believe where we need to be. And this is roughly 2 millimeters away from this, right? So because the distance of electrodes is also 2 millimeters. If we switch on this contact, in this case, the patient had 80% improvement of the movement symptoms. But if the electrodes are further away, this is again roughly 2 to 3 millimeters further apart, we're already at 25%. And then if we're really outside of the nucleus, often have a really poor response. So it really matters to hit the target, and it's really about millimeters. And for a neuroimaging perspective, this makes this so difficult because we, of course, have to reconstruct these electrodes as meticulously as possible if we want to make sense of them. And that is maybe a little bit different as in fMRI, where 2 millimeters are usually just a pixel and nobody really cares about the individual pixels, while here we really do care about them. And for that, we developed the toolbox that Felix already mentioned, LeadTBS, that is, uses pre and post-operative imaging. And the aim really is to make this as precise as possible. This is where I spent my money and energy the last 10 years to really try to make that as precise as possible and to reconstruct the electrodes and make them comparable across patients and centers. And one recent highlight that we could achieve was to use LeadTBS to detect or suggest optimal stimulation parameters that were then as good as the clinical team did. This was a real clinical trial, and I'm not going into details here, but just wanted to show that this really seems to work as a clinical tool. But you are all not deep brain stimulation researchers, so I thought you're more in the neuroimaging field. So I thought to maybe give you some reasons why you should maybe also care about LeadTBS, or why would it be interesting for you? I try to make this these few slides here. So first of all, what is really nice about the tool is that it comes fully packed with a lot of subcortical atlases. So if you ever need, atlas definitions in the subcortex of the brain, download it and have a look. It's all open, all in nifty files, can be used for anything as well. Really a lot of atlases, probably the largest collection of subcortical atlases there is. And we also have lots of whole brain atlases on parcellations built in there because we do a lot with connectomics. I think these you can also find elsewhere and probably have heard most of them, but they are also all in there as nifty files. It uses bits since the latest version, so it really integrates well with other tools. You could, do the normalizations in LeadTBS, for example, and I can come to that, and then use fMRI prep for, other things, and you can even trigger FreeSurfer from within LeadTBS. So it is a nice integrative tool. It has a command line interface as well, and so forth. Just for general visualization of stuff, it's also quite handy, I would say. So I'm just, made this video just to show this is the 3D viewer we have. And if you just drag in nifty files into this, you can visualize them like this. So in a 3D way. So, quite versatile. There's lighting effects and lots of things. So just as a visualizer, it could be a nice tool as well. It's in MATLAB. And this is a normal MATLAB figure, so you can plot anything on top. No restrictions, of course, there. So you can also show heat maps. This is just a nifty file shown as a heat map inside of the tool and customize it and so on. So maybe just for visualization, it could be nice. And then I think the most important point I wanted to show is that it really might be the ultimate normalization toolbox. And the reason is that we care so much about the millimeters, right? We really obsess about making this right. So you have all the SVM tools in there, all in, of course, in MATLAB, but there's also answer in a multispectral implementation where you can use T1s and T2 and other sequences in parallel to normalize. There's even the FSL and FSL tools in there, and they work in Windows too. We made that even though it's FSL, and then maybe the crown in the end is really this warp drive tool. So if you use ANTs for normalization, sometimes you might have the problem like down here in the cerebellum that you see that there's a mismatch between the normalized part and the template. And with warp drive, which is a tool we developed inside LeadDBS, you can manually refine these things. You can just drag with the mouse a bit like in Photoshop, and that works on the warp field level. So, you can look at, for example, now also different structures. This is the subthalamic nucleus that we are really interested about. If you, this is the normalized patient brain in this case, this is the template. So you really see some mismatch there if you fade between the two. This is an atlas now that matches the MNI brain. If we go back to the patient, we see there's a mismatch here, right? These should be in a different place, and then you can just use the mouse to fix that. Or you could even use a drawing tool where you just draw the outline, and it will find the closest outline of the atlas and snap it right there. So for anything that involves interest in hippocampal subfields or in cortex so layer specific fMRI, this is another video I made for you guys where I thought if you really want to overlay the fMRI blobs of specific cortical folds and you have a misregistration there, you can now use it and refine that, so to make sure that things really overlap. And I know this is quite specific, if you really focused on a specific area in the brain, but for that, it's a nice way to interact with normalization warp tools. And then last reason is that there is also an fMRI and diffusion MRI toolbox in there. It's called lead connectome. With that, you can pre-process resting state data and fiber tracking, whole brain fiber tracking with different methods. Works very easily. You just load it, press run, check the boxes, and it should work. And this is actually dating back to the first paper that Felix mentioned, that still the same code, of course, a bit updated over the years, but doing what we did back then. So that's, it might be that you hear LeadDBS, this is not a tool for me because it's something with deep brain stimulation, but it might be interesting even for general neuroimaging. But I want to now, of course, talk about what we do with it. If we're not involved in drinking or eating, we use LeadDBS for the general concept that we believe that we have to modulate a specific brain circuit that's broken or affected in a patient that would then define where we put the electrodes. And the same applies to non-invasive stimulation sites, right? We want to first know what's the circuit we want to modulate, and then where to put the TMS coil. And it could even apply to non-clinical things like, just symptoms, for example, risk-taking, or, these things. So it could be applied to anything. We want to know the circuit first, needs to be defined, and then we know where to modulate the brain to modulate that circuit. And the big question is, of course, how do we define these circuits? We can use animal models, but the translational value for clinical practice is really unclear. Do we have mice with OCD? Probably not. We probably have some with Parkinson's, but not with OCD. And so there's always this gap between animal models and humans. And then what most of you, I think, are using, functional imaging, very powerful technique, of course, but it remains sometimes unclear what is really therapeutic or compensatory. So if we would just scan 50 Parkinsonian patients and 50 healthy controls, see what's the difference, we are not sure what's the target, right? We would see that's different in their brains, but we wouldn't know which ones to modulate. And then even if we use electrophysiology or invasive electrophysiology, there's still this causality gap. So at the center here, we think of this slightly differently, and we essentially try to invert the concept. And we use the DBS sites and TMS sites to define the circuits. And we also use brain lesions as a third causal way of interacting with the connectome. So we do that by just localizing the electrodes, look at what are they connected to. This could be structural connections or functional connections. And then we compare across patients, right? We make this comparable and we see that some patients do well and some don't. So we contrast these things. And one specific method among like 3 or 4 that we developed is called DBS fiber filtering. And I want to briefly go into that because that's going to drive what I want to show you in the next slide. So usually we start with a full connectome or some set of fiber tracts that are interesting to us. So this is static. This is usually in template space. They're defined once for everybody. And then we go through them. We take each one out, a bit like we go through voxels in fMRI. We take one tract out in theory and in our mind, and then we see how does this tract now relate to different stimulation sites across patients. So these are just a few. Usually we deal with 50 or 100 or even more patients, but in this case, we would have 3 potential scenarios. So top responding patients that, where the DBS led to an improvement of a symptom, did all modulate this tract, and the poor responders did not, then we can kind of conclude, okay, this must be a tract that's associated, at least correlated with this beneficial effect. And then of course we have the opposite case where it's, just the reverse. All the poor responders modulated the tract, but the top responders did not. Then we can kind of, conclude this is probably not a good tract to modulate. So we should not do that in the future. And then the third option is maybe just it's unclear, right? There's no clear pattern related to this tract. So this tract doesn't seem to discriminate between top and poor responders. So we can probably just say, this is irrelevant for now. Let's filter it out. That's why it's called fiber filtering. We kind of want to see the tracts that matter and drive an effect. And again, this effect could be improvement of tremor, improvement of depression, of obsessions, but it could also be more risk-taking behavior in Parkinson's. It could be, anything. If we use TMS, this is widely applicable to any change that a modulation does to the behavior. And then we put these back in and we filter out the ones that were irrelevant. And this way we can select the tracts that seem to matter. This is just one example, in dystonia, another disease that we deal with. Don't want to go into deep details, but showing, for example, that in a specific subform of dystonia where only the neck region is really involved and affected, then the tracts that map to this homuncular region were relevant. And then in generalized dystonia, it's a different thing where also we have different palidothalamic tracts that were involved. So this is one example out of many of how we can filter the relevant tracts, and each of the tract has a correlation value in this case, or could be a different statistical measure, because of course we don't do this in a binary fashion as in my examples, but we quantify how relevant is each bundle. And I want to run you a bit more in detail across one recent study that we did with deep brain simulation for Alzheimer's disease. So the electrodes in Alzheimer's disease are implanted here into the fornix region, a bit in front of the fornix of the brain. And this was data from multiple sites across the globe, mainly Toronto. So the main lead of this is Andreas Lozano in Toronto, who's a neurosurgeon doing this as a, it's still under investigation, so it's not an established treatment here. But we were able to look at the tracts, at the data with our methods, and the electrodes here, if we applied fiber filtering, isolated specific tracts, including the fornix, but also the stria terminalis, which even received higher correlation values. And we can now, this was again sampling from a whole connectome, right? So any tract that's somewhat connected to this region could have emerged, but this is really what emerged. And if we just look at the correlation, this is a circular analysis of how much this set of tracts, would maybe explain the variance in the data. It's a nice fit. And we can also permute the data and see that this would not always be the case if we just add in, random values. But we can also go further and say these were now 28 patients from a first training set. So we define based on these 28 the set of connections that seem to matter for cognitive improvement in Alzheimer's disease. And now we can add, first we can do a leave one out cross validation. So we can recalculate the same thing, leaving out one patient, and then each time putting the left out stimulation volume on top of the tracts. So this is one example where the left out electrode modulated most of these tracts, and this one is one where it mostly hit missed the optimal tracts. And we see a correlation, between this fiber score, so how much of the red fibers are being modulated, and then the ADAS-Cog, so the clinical improvement following DBS. And this was now, in leave one out cross-validation, and in fact, we did the same with, multiple k-fold validations, quite robust effect. But we can also say, okay, we have more patients, 18 more, that were not even, seen by this model. And we can now always take exactly the same tracts. And we have one example again, where one of these 18 left out patients modulated the tract quite well, and one where it missed it. And it's again, a significant correlation in a really entirely unseen test cohort. So this is again, just showing the same for, the 28 patients, and then the test case, and then all patients together, always roughly finding the same network that seems crucial. And it seems to be anatomically the PAVE circuit that, has more or less precisely maps to it, seems to be the relevant circuit to modulate, to improve memory with DBS. And this is just for completeness. If we, we did this cross validation on the entire set in the end, using, leave one out, 10-fold cross validation, 7-fold cross validation, 5-fold and 3-fold, and really always remain significant. So this is quite robust effect. And then we also looked at the localized sweet spot of where exactly we think. So this is not looking at tracts, but more at pixels around the electrodes, and that explains similar amounts of variance. They are, of course, not independent from each other. And then we also used fMRI on a whole brain connectome to define which, functional connections seem to matter, and they were also quite robust towards cross validations. So maybe you believe now, or I could convince you that this could be helpful for clinical practice to help improve deep brain stimulation. But I think this crowd is more interested in, can we use this also to study the brain? Can we now look at this from a more neuroscientific angle rather than a clinical one? And I want to go back to the hits of this small nucleus in the brain that we mostly target in deep brain stimulation. It's called the subthalamic nucleus. It's a centimeter long. And what is so nice about it is that it receives input from the entire frontal cortex. So it is a bit like the thalamus, a core region that has a lot of connections, but it's smaller and it's, less complex in a way, but it does receive input from everybody in the front. And it is mainly a target for Parkinson's disease, but surprisingly, you can also use it and modulate it for dystonia, which is another movement disorder, Tourette syndrome, which is the tics disorder, and then even obsessive compulsive disorder, so a neuropsychiatric disorder. So people across the world have used this small nucleus to treat 4 different diseases that are quite different. And you would think, why is that? How can that be, right? How can we put electrodes? Is it all, scam, maybe? Is it all placebo or not? And it really is not placebo in this case. But why can that be, right? Why can we modulate this small region for these different effects? And so we sought out to look at that. Very big collaboration with lots of people involved. We have people in San Francisco and Shanghai that use the nucleus for dystonia. Of course, Parkinson is the most common one. We didn't have to look far for that, but then Tourette's is very rare. I think only these groups in the world did that. And then OCD is also quite rare. London and Grenoble had some experience and had data to contribute to look at this. And these are all the electrodes, targeting this nucleus. You see for different diseases, roughly the same spot. We know for OCD, we go more a bit to the front, but for the other ones, it's more or less the same target. And we then first looked at, okay, on the sweet spot level, where are the sites that map best to the best effect? So where do we need to modulate for each disease? And already you see a gradient, coming from the back in this more sensory part of the nucleus, which is the dystonia sweet spot, and then somewhat in between and even split across multiple parts was Tourette's disease, maybe as a more neuropsychiatric disease, and then Parkinson's disease is in the SMA receiving field, so in a premotor and also motor for more tremor symptoms field, and then OCD is quite anterior, right? So that's a difference, it's really a psychiatric disorder. So the more in the front, I would hypothesize that depression would be down here, and potentially we can also find some symptoms in Tourette's that would map here. But this is a bit the idea. We can see this gradient. Now, what about connections? If we just look at plain connections, how are the electrodes connected to the rest of the brain? That looks quite similar, right? This is weighted without fiber filtering, just connections of these. We already see this massive input to the nucleus from the entire frontal cortex. And if we now weight it based on clinical improvements, all of a sudden this pattern appears where specific subsections of this tract matter for specific diseases. And mapping them out like this, or even like this, we can see this beautiful gradient that follows the same gradient we've seen on the nucleus level that spans across the entire frontal cortex. And here we can even see the split of Tourette's, which I've seen, because many Tourette's patients have even obsessive compulsive symptoms as well. So there's certainly also some, let's say, psychiatric, in quotes, symptoms involved. But of course, we also have the motor tics, which are known to, where M1 primary motor cortex and also SMA are known to play a role. So to me, this is, applying a technique that was developed more for clinical practice and improving patient care to study the brain, to segregate the prefrontal cortex using some, different concept here. We can even think about, sorry about this. We can think about potentially information compression. So we roughly know now how distant these sites in the cortex are and where they map to in this nucleus. So we can think of this as at least spatial compression of the projections from the entire frontal cortex going to a centimeter long distance. So we can somehow perceive that this is some deep layer of a neural network that has a lot of convolution, a lot of integration of stuff. But then I mentioned OCD, but I don't know if anybody in the room knows OCD a bit, but I think most of you probably appreciate that it comes with lots of different symptoms, right? Obsessions, compulsions, but we also have a lot of comorbidity and depression or anxiety and other symptoms. So it's not a homogeneous disorder. It's a heterogeneous disorder. And so we can potentially zoom in further here and say, how can we maybe segregate this tract further into sub sections? And I want to start a bit with this tract to begin with, because even before this paper, we were quite involved in this specific tract. In fact, OCD is a disorder that's been treated with DBS, but not just in the subthalamic nucleus that we've been, but also in other sides of the brain. And that in a way screams for connectomics, right? That could be, it works everywhere, not everywhere, but it works in different sites, usually roughly the same way, comparably well. And of course, people thought long and hard where to put the electrodes. So there was, of course, hypotheses attached to it, but maybe it's all the same network. So we again collaborated with different sites started with Cologne and Grenoble. And this is now 2 different targets, and these are the main targets. This is one in the anterior limb of the internal capsule, and this is the well-known STN that we've already talked about. If you just look at the connections again, you know, very different profile. STN, as mentioned, receives this whole input from the frontal cortex, but the ALIC is you know, more anterior, so it's you know, it doesn't involve the motor cortex, for example. But if we use fiber filtering in each of these cohorts alone, the same track that I've already shown you before seems to appear, right? The same one associated with specific improvements of symptoms. And this was independent from each other. Now we can even calculate the track based on the ALIC patients and then put the stimulation volumes of the STN cases on top of that and say, how much do they hit this ALIC tract? And does that correlate with clinical improvement? And it does. So this is the Y-box improvement of obsessive compulsive symptoms. These are the fiber scores. We can do the reverse as well. We now calculate it just based on the subthalamic nucleus cases. These are the stimulation volumes of the ALIC cases. And again, this correlates quite nicely. So the more they hit this tract, the better. You might think this is probably fishy. This is too good to be true. You know, can't be true that it's so, you know, nice. But the story goes on. So we did look at in the literature, where did people put electrodes, and it already clusters quite nicely around the tract. These are a bit more below it, but the electrodes stick up like this, right? So often the targets are defined a bit, you know, lower than the actual stimulation sites. And if we measure the distance between each site and this tract and the improvement reported in the study, this correlated. Just 6 data points, so probably not, you know, convincing. Still nice lead. Now we did present this data at conferences in, you know, 2019 or so, and the London team was there. We had a rotational student from Madrid at the lab at the time. And these were additional cohorts, unseen. This was already as a preprint out there, you know, there's proof of that online. We were not able to, you know, change this tract anymore with the new data sets, but we used it to predict them, and it did work. And then the nicest thing is once you publish it, when other people replicate it, right? So this is 10 additional patients from New York that Helen Mayberg's team where they looked at this again. They also, you know, looked at how well does the stimulation volume hit this tract, and, you know, significantly correlated. The more you modulate the tract, the better. And these were in fact 4 replications 6 months after the publication from different groups that all showed this really seems to be true. And it seems to be true even across, you know, targets originally used. And this one is even for Tourette syndrome, so obsessive compulsive disorder. Symptoms in Tourette's syndrome also worked on a large cohort. So this really seems to check out. And the last thing we did here was to do it in a blinded fashion. So Samir Sheff in Texas had 10 additional patients. They sent us just the neuroimaging and the stimulation parameters. So we had no clue about the patients, you know, nothing about the clinical symptoms and so on. And we ran it through this tract model and predicted the clinical outcomes. And we sent that back to Texas. And that was a nervous email to send, because we really had to say, this time we put our nickel down. This is, you know, how we think they would do. And sure enough, Samir Sheff's team would know the actual clinical outcomes and could then correlate them. And of course, both teams were quite excited that it did work. And this is, again, just 10 cases, but with, you know, each time, each replication, I think this really seems to be a robust finding. This is just showing the same track originally published, and then now on these 10 new patients, really same story seems to appear each time. And it also makes sense, because this track does, you know, link different therapies that we know work. So we know from the history that the DACC region, where this tract originates from, has been, you know, lesioned quite a bit with success in OCD. We know the capsule itself has been lesioned with conventional methods, with gamma knife, with pus, and that seems to work. We know DBS down in the STN seems to work. DBS here does seem to work. And even with non-invasive sites, there's even an FDA approval for TMS, also targeting the same DACC, DMPFC connection that seems to matter. But I wanted to talk about the heterogeneity, right? So I started off saying OCD is not a homogeneous disease, a disorder. So again, we, wanted to go all in on this and asked all our friends and said, hey, we need more data. We want to look at subsymptoms here. This is ongoing work, probably one of the largest we've, done so far with lots of electrodes, work led by Barbara Holunder. These are all just the active contacts or the stimulation sites in the subcortex. And this is the global OCD track that I've shown you multiple times now. And if we look at specific subsymptoms, we see a segregation. Here you can see it at best, that, again, similar to what we've shown before, that specific subsections of this ALIC bundle seem to map most to improvements in obsessions, compulsions, anxiety, depression, and also global functioning. Can do the same with Parkinson's disease, where also we have, different symptoms, tremor, bradykinesia, rigidity, and so on. So we want to zoom into Parkinson's disease now. This is, again, this bigger model, but here we can also sub-segregate the tract into different symptoms like tremor, bradykinesia, rigidity, and axial symptoms. And you can really appreciate they map to slightly different parts of the nucleus. The bradykinesia one maps to the more medial surface of it, or medial aspect of it. And looking at it again, we can now build a model to predict specific symptoms in Parkinson's disease. What is interesting for the nerds out there is that the axial symptoms seem to map to the brainstem, to the PPN nucleus, which is known to be involved in axial, like in gait processing specifically. And then the green tremor tract seems to map to the cerebellum, which is also a very well-known connection, that dentatorubro-thalamic tract. This is on the cortical surface. We also knew since ages that tremor would map to M1 versus the other symptoms would rather map to the premotor cortex. And Hassler, famous German physiologist and anatomist, he published on that in the 60s, exactly the same findings, where the M1, like the tremor connection from the cerebellum, would map to M1, and then the more slower symptoms like bradykinesia and so on would rather map to the premotor cortex. This is, again, this cerebellar connection maps to tremor, same shown back in the day from Hassler, coming even in the same color, coming from the cerebellum to the M1 region. And then axial symptoms, I've mentioned that this specific connection to the brainstem kind of makes sense to people in the DBS field because we have targeted this nucleus down there for freezing of gait in the past as well. So I could imagine that potentially with this concept, we could move from what we currently do, which is one stimulation site to hit all symptoms, to a much more fine-grained stimulation where we could really target specific subsymptoms for specific patients that suffer from specific symptoms in the disease, and do the same for OCD and for Parkinson's and so on. Could even apply different frequencies. Doesn't make too much sense probably to most of you, but for gait, we know that lower frequencies are sometimes better, and for tremor, we know that higher frequencies are better. And then I want to conclude with some non-motor symptoms, just to show in Parkinson's disease, we also have lots of different symptoms that are affected, including apathy, depression, impulsivity, also cognitive decline, and so on. So we can map these two tracts as well. And since there are so many, we chose a different strategy now. We mapped them first to PCs, the principal components, using PCA. And then each of these 4 components that made most sense to us mapped to a specific tract. And now we can say, okay, if we now have new patients that modulate a combination of these 4 PC tracts, can we maybe deflate it again and predict their individual symptoms based on the combination of the modulated tract? So in a way, we map the PCs to specific tracts, and then we can reverse the PCA again and show improvement in specific clinical outcomes. And that at least on a within sample level seems to work, which is already nice, but it's quite circular in this case. But it already shows us that we can map things to specific 4 tracts and then deflate the information again to go to specific symptoms, and of course needs to work in out of sample data, and we have good hints that this will also work. So with that, I hope that we can kind of come to a personalized deep brain stimulation. And I want to remind people again, this same concept would be also applicable to any form of neuromodulation, right? So we can do the same stuff with TMS. We can also use functional connectivity instead of structural connectivity, which I focused on. But the idea to map symptoms or behavioral changes to specific networks, once we have them in MNI space, to then in individual patients or individual goals to map specific find the optimal site to stimulate based on what we want to modulate. And to wrap up, just a few slides, what's next? So I think my lab has now published lots of these maps of optimal connectivity, and again, I've shown a lot of tracks before, but we also have quite a few functional MRI maps where these come from different papers, but we now need to find a better way to connect those dots and essentially create a taxonomy of symptom networks across the brain, because we also want to make connections here. For example, we have depression in Parkinson's disease shown here, but we also have, of course, depression in major depression, and we have depression in epilepsy, and we have depression in OCD. And there's first evidence in a paper by Shan Siddiqui that they all map to the same network. We also have tremor and essential tremor, but we also have tremor in Parkinson's disease. And we have a paper that shows that these map to the same network as well, right? So we can or cognitive decline in Alzheimer's and Parkinson's, we kind of want to show that these are symptom-specific networks and not so much disease-specific networks. And then maybe less interesting for you guys, but still I wanted to show it because it's a nice video. We also want to transfer this more into the operation rooms to actually move this technology of finding optimal targets into the surgical room. This is a software called LeadOR that Simon Oxenford developed in the lab, and we can record while we go down into these nuclei, and we see the firing patterns here. And we also see, the patient-specific anatomy. But we can now bring this together. We can kind of fuse the recordings with the anatomy. And that is shown in the next step where going down, this is the firing rate mapped to where we are in the brain. By the way, this is a histological slide from the big brain. So we can also, add atlases here. We have all together, and we know while we go down where we are. And this is just showing also that the firing rate matches quite well the anatomy. This is a lead that's outside of the STN, that's next to it, and this is inside of it, and it really matches quite well where we see the firing rate and where we see the imaging. And that's again this, our obsession of precision, of trying to make the millimeters really work, because if not, everything we do is senseless. So this is the next step in the surgery. Again, we then test simulate. We would also see which tracts are being modulated in each point in time. And this blue one is the internal capsule, which leads to side effects. Before it was the hyperdirect pathway. So we can again correlate what we see in the operation room in terms of symptoms and side effects with what we see in the computer in our prediction of what we would do. And this is all done in real time. So to sum up, I hope some of you at least might find it interesting to maybe download LeadDBS and look into it. Could be really interesting just to normalize brains and potentially integrate with fMRI prep or whatever everybody uses these days. And if it's just about the atlases or visualization tools, would be great. Happy to help in case anybody wants to look into it. And maybe I could convince you that we can use these invasive neuromodulation ideas like deep brain stimulation to study the organization of the brain to some degree, and then also to map symptoms across diseases and map symptoms improvements across deep brain stimulation targets. And I want to briefly mention that we will be at OHBM quite fully fledged this year. We have 3 invited talks, and I have a keynote, and then since I have fond memories of going to my first OHBMs with Felix Lab, I thought to bring this up. We'll have an Airbnb, and I hope we can meet up there, and if some of you go to Montreal. And with that, I want to thank you. I currently really have 2 labs because I'm ramping down in Berlin and ramping up in Boston. So these are the 2 group photos here with 1 double, I think. So he moved with me, Clemens, and lots of people I have to thank to show this, of course. And I hope I had enough pictures already of the trainees that did all the hard work and the collaborators and so on. But with that, I want to say thanks for the attention and thanks again for the invitation. And of course, happy to take any questions.